Hello and welcome to today's lecture. So today we will briefly review what we had discussed in greater detail in last two classes, which is correlation and regression, and discuss about some aspects of fitting data. Okay. So just to summarize what we had done in the last few lectures, we had discussed how to plot bivariate data x and y, and then that from the plots can you get some estimate of what is the extent of correlation between these two variables x and y. Okay. So in this particular case, we can draw a curve something like this, we can draw a curve which is horizontal, so on and so forth. Okay. So to quantitatively determine the extent of correlation between two variables x and y, so the correlation we compute the correlation coefficient. So the correlation coefficient is written by either rho or r, and it is okay. So and it is defined by s xy by sx times sy, where s xy refers to the covariance, and defined by s xy is equal to summation of xi minus x bar into yi minus y bar whole divided by n minus 1. Okay. And we also discussed that the correlation coefficient is actually bounded between -1 and 1. So for two variables which are perfectly correlated, for example, if we take x equal to y, in this case my correlation coefficient is going to be +1, versus if I take x equal to -y, okay, so this is my x, this is my y, okay, x equal to -y, then you have this particular value, okay. So minus x, yes. So this is y is equal to minus x. So in this particular case, my correlation coefficient is going to be -1. Okay. So however, if your data points are all scattered, okay, data points are all scattered, as in this case, let us say, when you compute the correlation coefficient, you might have, so for a line which is perfectly horizontal, your correlation coefficient will come to be either perfectly horizontal, or let us say if it is perfectly vertical like this. In both these cases, my correlation coefficient will actually approximately turn out to be close to 0. Okay. Now one of the immediate follow ups of correlation is regression. Okay. So if you have a set of points x and y, okay, like this, can you estimate y given one value of x or vice versa? Okay. So here the objective is to approximate these data points by some curve, maybe linear, as is as seems from the trend from these particular points, and can we in case of linear regression, okay, in case of linear regression, I want to approximate this data by some equation like a plus bx. Okay. So for doing linear regression, right, so once again you have x and y which are plotted like this, okay, and I want to approximate by this particular curve, which is y is equal to a plus bx. So I had derived in few 2 or 3 more back lectures that you can find out the value of a. So you have from this equation, I get one equation which is y bar is equal to a plus bx bar, and the other equation which I get is b is s xy by sx square, and this will give me a value which I can also write it as rho times sy by sx. Okay. This is the value of b, and once I calculate the value of a is nothing but y bar minus bx bar. Okay. So this is how you calculate a and b. Okay. So let us take one example once more and do one more thing. Okay. So let us say the first step is of course to find out the values of a and b respectively, but the next step is to ask, is this equation a good enough fit for this particular experimental data, right. So let us do one particular example. So let us take the following values, x is 1, 2. So let me make a data like this. Okay. This is my x, this is my y. So x is, let us say, 1 1, then you have 2 2, okay, 3 2, 4 3. Okay. So, what you can clearly see, so if I can actually add one more point, or maybe what I do is 4 2 and 5 3. Okay. Okay. So, my data points look something like this. Okay. So, my points are once more, okay, x and y 1 1 2 2 3 2 4 2 5 3. Okay. Okay. So, I know, so I have to calculate S x y and S x, right, to in order to be able to compute B. Okay. So, for these particular values of x, I can calculate. So, S x y is given by summation x minus x bar into y minus y bar whole divided by n minus 1. Okay. So, x bar comes out to be 3, y bar comes out to be 4 and 6 10, y bar comes out to be 2. Okay. Okay. 1 2 3 4 5 1 2 2 2 3. Okay. So, in this case x bar is 3 is minus 2 into minus 1. In case of 2, it is 2 minus 1 into 2 minus 2 0. Is 3 is also will give you a value of 0, 4 will also give you a value of 0. In case of 5, you have 1 into y minus 1 bar minus 1. Okay. So, in this particular case, I can then find out the value of S x y is going to be minus 2 minus 1 divided by n minus 1 is 4 is equal to minus 3/4. Okay. And S x is S x square is going to be x minus x bar whole square, x bar is minus 2 square plus 2 minus 1 square plus 3 0 square plus 4 minus 1 square plus 2 square by 4. Okay. So, it is going to be, okay, it is going to be 4 plus 1 5 10 by 4. Okay. So, B turns out to be S x y by S x square is equal to minus 3 by 10. Okay. And A comes out to be y bar minus B times x bar is equal to 2 minus of minus 3 by 10 into x bar is 3 is equal to 2 plus 9 by 10 is equal to, okay, 3 plus 9 by 10 is turns out to be 29 by 10 is approximately equal to 3. Okay. So, my equation is y is equal to approximately 3 minus 3 by 10 into x. Okay. Now, I want to find out, okay, so for each of the values, okay, so let us say okay, 1 2 3 4 5 1 2 2 2 3 predicted value is, so I can actually, maybe I should write 29 by 10, okay, x equal to 1, 29 minus 26 by 10, 2 6 23 by 10, okay, for 3 is 9, 2 4 5 okay, 4 to the 12 and 5 15 14 by 10. Okay. So, I can clearly see that for these two values, it is doing a reasonable job. For 1 and 5, it is really doing a very bad job, and the reason is obvious. Okay. If I were to draw the points again, okay, see these are your points, and the line it has been drawn is roughly y is equal to 3. Okay. Am I doing plus b? Okay. So, your line, so this is doing a very bad job for these two extreme points. So, I can compute the error, which is the difference between the actual value of y and this deviation. So, this comes to 16 by 10, this comes to minus 3 by 10, this comes to 0, this comes to 3 by 10, and this comes to 16 by 10. Okay. This is minus 16 by 10. Okay. So, the way to estimate the goodness of fit of this data is to compute is, so what you typically have is, if you do it in Excel or Origin or any of the standard statistical softwares. So, in additional to this equation that they write, they also write a value of R square. This R square is a measure of goodness of fit. Okay. So, this R square is given by the expression R square is equal to 1 minus summation of E square by y minus y bar whole square. Okay. So, we have calculated the errors. Okay. We have so, if I do the particular values, okay, we have error, and I want to plot y minus y bar. So, I have 1, 2, 3, 4, 5, y is 1, 2, 2, 2, 3, error predicted. So, error is minus 16 by 10, minus 3 by 10, 0, 3 by 10, and 16 by 10, and y bar, y minus y bar. So, y bar I know is equal to 2. So, y minus y bar is minus 1, 0, 0, 0, and 1. Okay. So, my goodness of fit in this case is equal to 16 by 10 whole square into 2 plus 3 by 10 whole square into 2, and y minus y bar is 1 square plus 1 square. Okay. So, this is a significantly large number, 16 by 10 here. Okay. So, I will get a value of R square which is reasonably low. In other words, this is not a good fit. Okay. So, you can compute the R square value. So, for whenever you do the calculation, you should also compute the R square value, and then come to a conclusion if your line is a good representative of a data. So, if for a very good curve, so let us say for if we have a curve which exactly are fitted to the point y is equal to s. So, in this case you will get a value of R square is equal to 1. So, in best case situation, if you have a perfect fit, R square should be should return you a value of 1, versus if it is a bad value, you should get a very R square value which is very low. Okay. So, this tells you that this is how we compute the R square value or the goodness of fit of the data. Okay. Now, one of the obvious usage of doing linear regression is to use it to estimate a value of y given a value of x. Let us say, okay, we have this particular, so we had this data based on which we fit this line, okay, and we want to know what is the value of x. So, let us say this is 1 and this is 5, and I want, so this is my x, this is my y, and this is a particular equation, let us say 2 plus 3x. Okay. So, I want to know the value of x, I want to know the value of y given a value of x. Let us say I want to know the value of y at x equal to 3.2. So, what I do is I simply plug in the value of x and get the corresponding value of y. So, this is called interpolation. Okay. Interpolation means that the point you are trying to get an estimate. So, estimate is obtained in the range where the data is fitted. Okay. So, because the range over which I was fitting my curve was between 1 and 5. So, when I put an x equal to 3.2 value, it falls within this range. So, this process is called interpolation. Okay. And this, so let me give you one very common use of interpolation in biology. So, for example, if you want to do, let us say, a western, right, you have 3 samples A, B, C. From these 3 samples, you have collected the total cell lysate, okay, lysate, which is the content of all the proteins present in these cells cultured under, let us say, cells which have been cultured under conditions A, B, C. So, and I want to ask the question, how does the expression of protein X, so X is a protein, okay, how does the expression of protein X vary as a function of the conditions A, B, C. So, what I need to do is, as a first step, if I want to run a western, so in western what I do, I want to load proteins of, so I want to load total proteins Y, let us say, of each sample A, B, C, and then get some bands in electrophoresis using page, and I want to develop these bands and ask the question, how is the expression profile. So, this is the band you get corresponding to Y, and I want to know what is the, how is Y variant. So, one of the critical steps is to ensure that so this is your protein X band, and you want to know how X is varying across different conditions. Okay. So, these are your conditions A, B, and C. So, in order to make sure that you are comparing X as a fraction of the same equal amount, you want to make sure that the same amount of protein Y is loaded across all the conditions. Okay. So, which means that you want to know what is the protein concentration of each of the samples A, B, and C. So, the question to begin with, you want to estimate the protein concentrations of A, B, and C. Okay. So, how do you do it? So, what you do, you use something called a standard, let us say standard, okay, and typically people use BSA or bovine serum albumin. What you do is you prepare stocks of various concentrations, let us say 0.5, so on and so forth. Okay. So, you use your protein samples of known concentrations. So, if you know that the highest concentration of any of your samples A, B, C is going to be less than 1 mg per mil, what you do is you prepare protein stocks. So, these are all stocks of varying concentrations. Okay. You prepare these stocks and you get the OD measure from a spectrophotometer. So, what you will have are these points which will give you these data points, and to this you would fit either a line, okay, you would fit a line to get what is the estimate of your sample. Okay. So, to get an estimate of what is Y is equal to A plus Bx is what you will obtain. You will obtain the equation corresponding for these data points which are prepared with stocks of various concentrations. Okay. So, this is your step 1. In the next step, what you do is, let us say you take your protein samples A, B, C, and you use the spectrophotometer to take the OD value. Okay. So, in the background of this, what you have is, let us say you have the Y value. So, let us say this is for sample A, this is for sample B, and this is for sample C. Okay. These are your OD values corresponding to sample A, sample B, and sample C. So, how we do backtrack and find out the concentration? So, if you know the equation, you can invert the equation to find X is nothing but Y minus A by B. Okay. So, because you know the OD value, which is your Y, so Y is your OD value and X is your concentration. So, given the OD value, you backtrack using this equation to find out the exact concentration of A, the exact concentration of B, and the exact concentration of C. Okay. So, this is how it is widely used for interpolating data and to finding out how much protein you should use, so that in order to assess that across condition A, B, C, what is the expression changes in the expression level of protein A. Okay. So, this is the way procedure of how you make use of interpolation to find out a given value. Okay. Now, Interpolation is reasonably straightforward. If your data points do fit a very clean line, then the fit is nice and your protein estimates are good. What is more difficult is to extrapolate. Okay. What is extrapolation? Okay. Let us say you are probing the binding interaction between a ligand L and its receptor R. Okay. So, when L binds to R and you want to find out the binding strength of this interaction. Okay. You want to probe the binding strength of this interaction. Okay. So, experimentally one of the assays used, so if you want to measure actually in terms of forces the binding strength between L and R. So, what you want to know is how much force is required to tear apart a bond which is formed when L and R. So, when L binds to R. Okay. So, you can use any experimental assay, let us say for example, atomic force microscopy is one such assay which can be used to probe this. So, the critical problem here, so you want to estimate the binding strength of one molecule, okay, but it is nearly impossible to estimate the the binding strength of one molecule, because this measure of let us say 10 piconewton force is way below the resolution limit of your system. So, what you do is let us say you take a surface and you put down known concentrations of your receptor R. Okay. You put known concentration of R, okay, and then what you do is you take a tip and you vary the concentration, you functionalize this AFM tip with your protein L, okay, and you vary the concentration of L. So, essentially you do the experiments between L1, L2, L3, so on and so forth. So, let us say I begin with 1 mg per mil concentration, L1 is 1 mg per mil concentration, L2 is 0.5 mg per mil concentration, okay, and so on and so on and so forth, and I keep reducing the concentrations. Okay. And let us say, so I have no way of knowing exactly how many bonds have been formed when this L1 number of ligands bind to this particular things, because you are operating at a macro scale where you only have control over this concentration which is 1 mg per mil. It does not, you do not know exactly how many molecules of this particular ligand are there on the tip. So, it is an estimate, it is an approximate, you know the scaling, but you do not know the exact value. So, how will you go ahead and do it? Okay. So, let us say I plot the unbinding force. Okay. So, this is my concentration axis and I plot my unbinding forces, okay, that I measure using an AFM. Okay. So, at a very high concentration, logic would dictate if at L1, which is my highest concentration, I get this particular force, that an L2 which is at half concentration, on an average I should see a lower force. Okay. And so, this curve should keep on reducing, should reduce, and then for some concentration, okay, for below a certain concentration I begin to see a plateau. Okay. So, I begin to see a plateau. So, if I begin to see a plateau, then I know, and let us say this is my lowest concentration which was L10. Okay. So, till L10 I begin to see a plateau. So, what this means is in this range of concentration, it is likely that only one bond is forming, okay, because if I reduce the concentration further, I only hit a value of 0 and nothing. Okay. Below this concentration I only hit 0. So, this means that I am operating very close to either one bond or two bonds. Okay. So, using this, if I know at this concentration I am getting a plateau, I know that below this, so this is roughly mimetic of the binding strength of a single bond. So, this is how I can make use of extrapolation, even though I do not have a way of controlling the interaction between a single bond or forming a single bond, but by doing these experiments and by extrapolating, it might be possible to operate at a level such that this force can be estimated. So, this is extrapolation, but there are of course lot of fear. So, the problem with extrapolation is you are still making. So, let us take an example. Okay. Let us take an example. Okay. Imagine my curve is something like this. Okay. Now, and this is my x, this is my y. This is the regime within which I have collected my data. So, and I want to know the value of y at this particular value of x. So, do I assume that the curve is doing something like this, or the curve is actually saturating beyond this point. So, this is what makes the process of extrapolation somewhat more complicated, and many times you might actually get lot of errors as if you pre-assume the nature of the function. So, the challenge is really to use the appropriate function to fit your data, so that when you extrapolate this particular function to get the estimate of y for a given value of x, it is a reasonable estimate. With that, I conclude my lecture for today, and I hope you have gotten an idea of how we can make use of linear regression to fit data and to make use of it to interpolate and extrapolate. Okay. So, if your data is if your data is very erroneous, then if your data has wide scatter, in this case extrapolating can give you large errors. Okay. Interpolation is reasonably okay still, but still, as I have drawn in this case, if I get a line like this at this point, you see the amount of deviation from this line. So, you might still be the estimate might still be very erroneous. With that, I thank you for your attention, and I look forward to next class.